All right, so I think this is a great thing that we're doing here, you know, getting more familiar with one another. I hope you notice in my email, I said the FMEL family, although I've only been here about 6 years now. I do feel like this place is my second home and all of you are part of my extended family. And I've been fortunate enough while I've been here to work with, you know, amazing people. So, you know, I want to take the time to thank my lab sincerely, you know, your dedication and interest and focus on stuff. You guys make it happen. You know, I just sit at the computer and at the phone and answer your questions and stuff. So my pleasure. So I'm not going to talk very much about myself. So I really love mosquitoes, you know, just love them. And really interested in the diversity of mosquitoes that are out there, what they're doing in nature from every aspect. And then through trying to study them, I've had to develop some new kind of technologies to collect some of the species that aren't Aedes aegypti or Culex quinquefasciatus. And so a focus of my program has been developing new sampling strategies for mosquitoes, typically those that aren't the major disease vectors, but those that are vectors. And so in no particular order, I think it's just the order of people who gave me their presentations, we'll just get started.
Thank you. As a lab technician, I have been working in many areas. So I'm responsible for the mosquito colony, involve many species and population. I also help in the ecology lab, in the molecular lab, and in the field.
Excuse me.
For the past years, I have been focused on mosquito trap created by our lab using low technology approach to vector sampling. So the next few slides, I'll be presenting very briefly some of these field tools. Here we have a gravity trap, a light trap, and a resting trap. In the capture chamber of each trap was designed to accept a honey card to collect mosquito saliva for our virus detections. This is the red cap trap. It's a very inexpensive trap, and it was designed specific for Aedes aegypti seeking a blood meal. In the capture chamber of this trap, it's also designed to accept a honey card to collect mosquito saliva for arbovirus detection, and also to provide sucrose solution to increase the capture mosquito survival. This was a modification of the intake funnel of a BG trap net to accept a falcon tube with a preservative pigeon. And this novel allowed us to collect for 1 week blood engorged females in the field for blood meal analysis. We also attached a timer to the battery to control the trap operation and also increase the battery lifetime. This is a very low cost light trap. This was we brought this trap to the course in Brazil last year and was the highlight of the course. The students were very excited about it. It's made out of like a milk jar, as you can see, and a plastic tray and all the inexpensive material. Here we have a homemade chemical lure. This lure consists of 2 ceramic spheres, and in each sphere we add 1 base solution and an acid solution, and we place on the bottom of the red cap trap, but also we test with the BG trap. Carbon dioxide is another lure, very common used in many traps around the world, in forms of dry ice or gas tank. However, it's very expensive. It's hard to transport in the remote areas. It can be unavailable. So in our lab, we produce CO2 from the combination of common acid and common carbonates. We design a container, and in the top we place the acid solution, and on the bottom the carbonate, and a timer which is optional to control the delivery of CO2, and a hose to direct the output of CO2 in different traps. Can be used in the passive trap, light trap, the BG trap, the red trap. This is a resting trap. This is a very inexpensive trap as well. The body trap is made of plastic and wire. It's very easy to transport. for the remote areas, we don't need to use aspirator or battery or lure, and the specimens will in good condition. I put in here a picture of the CDC backpack aspirator, just to mention that it's a great tool. However, it's very heavy to carry. So in our lab, we developed the handle aspirator in this case, we were aspirating Culex quintofasciatus, Aedes albopictus, and Aedes aegypti from the resting area. I would like to point out here the mouth aspirator that also developed by our lab so I could reach the native bromeliads in the higher position. We also sampling the exotic bromeliads, and we are looking larval population, exotic and native bromeliads. So in summary, I just would like to say that we have been working with simple and adaptable technology to support studying ecology of vector-borne disease. And thank you for your time.
Hey everybody, my name is Theo Black. I started working in Nathan's lab in September on the Cervid Health Research Initiative, and I'm pursuing my master's degree. So what Cherry does is we're trying to protect captive commercial deer populations in Florida, and one of the main risk factors for these deer farms is a disease called hemorrhagic fever disease, which is spread by the bites of Culicoides. So one of the challenges of working in this system is that there are a lot of Culicoides. I may not have to tell you that, but there are truly massive numbers of these that are coming out of these habitats. And a lot of control methods don't work as effectively as they do in mosquito populations, for instance. Adulticides, fogging have not been proven to be very effective, even in small sort of contained areas. And larval habitats and the driving factors within them are also largely not well understood, especially here in Florida, where we're working with multiple species and some more complex habitats. Many of the sort of successful larval control methods have been around, dairy waste ponds where the Culicoides are living in pretty narrow ranges. But here, they're all across the environment. So here's our main field site in northern Florida in the Panhandle, and all around here, this is just wet. In the Panhandle, you have this like very dry pine savanna, and you descend in a couple of meters, it's completely swampy. So as a new person coming into this environment, you get down there and it looks wet. And so you're trying to find where these very small organisms that live in moist soil are located, and it's hard to pick out where they might be specifically. And it can be more difficult than at first glance. You can't just put down anywhere. So I'm sort of investigating a couple of different methods to try and characterize where these larvae are, where they're concentrated. We're looking into different emergence trap designs. Nathan really likes umbrellas. And I've also tried out this noble instrument, the sod plugger, which allows us to take a soil sample and put it into a rearing chamber. So I got to try this out this summer. I took about 33 of these cores in the field to just kind of see what was going on, get my feet wet. And, this is very preliminary, but one of the takeaways that I'm getting from early data is that it can really matter where you put down. So each one of these dots represents a core that I took this summer. And there can be pretty big differences, even within the same area. I'm just out there trying my best to find where I think the Culicoides are, and it's not as easy as it looks, I guess. So that's this type of thinking is going to be driving my thesis research going forward, and hope to be sharing some of that, more of that with you guys soon. Thank you.
Thanks. So I'm just going to be talking a little bit about my PhD dissertation research today. I'm primarily studying the ecology of Culex Melanoconion vectors of Venezuelan equine encephalitis virus. And my primary research interests include optimizing surveillance, kind of relating vector host associations with habitat characteristics, and then using these to elucidate as a whole, as an overall goal, elucidate transmission cycles of mosquito-borne pathogens through vector host associations. So I've mostly focused on optimizing surveillance techniques for these vector species so far. And this system that I work in primarily is Everglades virus, which is endemic to Florida. And it's an enzootic subtype of Venezuelan equine encephalitis virus with transmission cycles, including Culex CDCI, the native vector, and the primary reservoir hosts, such as cotton rat and cotton mouse. And a kind of a ongoing question with Everglades virus is whether or not there's a potential for it to mutate into epidemic strains, because a lot of the other subtypes of Venezuelan equine encephalitis throughout the Americas have the potential to do this and can affect human and equine populations. And so this is a particularly important question now because of the recent introduction of Culex panicasa in 2016, which is discovered by our lab, which is the vector of VEE1D in Central America. And so the two primary organisms I study are Culex CDCI, which is native to Florida and is a competent vector of Everglades virus, and Culex panicasa, which is not native to Florida. It's native to Panama, the Canal area, and it is a competent vector of Venezuelan equine encephalitis subtype 1D, but we don't know yet if it is competent for Everglades virus, but it probably is because subtype 1D is very closely related to Everglades virus. So one question driving my research is how might adding this new vector species affect the cycle of Venezuelan equine encephalitis, or Everglades virus in Florida. So adding this new mosquito species, we're currently not sure what it's feeding on, but that's kind of in the works right now. It could be feeding on rodents, just like Culex CDCI, or wading birds, both of which have been known to been fed on by Culex panicasa in its native range. So either way, whatever it's feeding on, they're probably putting humans into closer contact with Everglades virus. In order to kind of figure out what they're feeding on, I this past summer conducted a trap comparison to help to figure out the best traps to use, not only for Culex CDCI and panicasa, but also just for specifically collecting blood engorged specimens of Culex panicasa. So this is important because the traps you use in studies often depend on your objectives. So whether you're doing blood meal analysis, viral detection, just general surveillance, or even the type of species, which species you're looking after. So if you're kind of trying to find 80s, you might use a VG trap. So I used a variety of different traps down at a field site in South Florida where Culex CDCI and panicasa coexist. And I used both commercially available traps, the BG and CDC light traps, as well as some aspirators, resting shelters, and our novel mosquito drift vents to capture some of these mosquitoes. I really wanted to find, like I said, the blood engorged Culex panicasa, and the reason for doing this comparison was because while we primarily use the resting shelters to collect blood engorged Culex CDCI, it was really interesting to find that Culex panicasa really doesn't go to the resting shelters at all. Even males, I've only found about 2 males throughout my studies in these resting shelters. And so in order to study the host associations, you need to figure out what kind of trap to use. And so just a real quick overview of some of my results so far. indicate that aspirators might be very important for collecting blood engorged specimens of the species. And I've even caught some in our mosquito drift nets as well. And so I'll be talking a little bit more about my results from this study, probably at the Byler Symposium. So that's all I have for today.
Hi everyone. Well, my dissertation is related to the dynamic of hemorrhagic disease, how we can connect entomology, disease ecology, and community ecology. I will talk about the system that he already introduced us. One of my main research interests is how parasites are maintaining the ecosystems. The definition of where are the boundaries on the ecosystem is sometimes artificial. So for that reason, the ecosystem terminology or a framework is pretty interesting to try to understand how we can define the borders or the boundaries of the disease. So in the case of hemorrhagic episodic disease, we have several viruses with several serotypes and also strains that interact with multiple vectors and multiple hosts. And for the ecosystem Florida, we already don't know exactly where are that vectors and where are that hosts. So the idea is to try to integrate all the ecological information that we have, but with a lot of unknown parameters related with the biological aptitude of these vectors and hosts. So my research questions are around how the dynamic of Culicoides community and the interaction of the Culicoides and hosts could be used to inform about the dynamic of HD. Well, the first thing that we have been doing is try to understand how different traits of the host related with the host competence or how they are using or exploited by Culicoides as a blood meal can be used to redirect the management operation to reduce or prevent hemorrhagic disease. So when we are talking about the network, it's easy to understand that we have two communities, a community of hosts and a community of vectors or Culicoides in this case, that are connected throughout links, and the links are the blood meals. So when we have two communities that are interacting, the networks are pretty cool tools to try to understand that complex interactions. So as we don't know exactly what is the vector competence of each of that species, and also we don't know that about the host, we try to simulate how the blood meal interaction could be related with a virus transmission pathway. So if we make different simulations to try to, for example, immunize, give a vaccine, or to control the amount of Culicoides that can feed, for example, in an elk, we can reduce hypothetically the number of pathways that the virus can have. So the idea was to make different scenarios when we try to make control between a species related with the host competence, the degree of the interaction, that is just how many blood meat can we take for any host, and how a host can connect different parts of that network. And it's very interesting that the interaction are pretty more important than the aptitude of the host to transmit the virus, or to sustain the virus in the ecosystems, and that can give some information of where will be we focus to try to make decisions to control the disease. The second issue that was important for us is how that impressive explosion of Culicoides, when several species are involved, could give some idea about the dynamics. Historically, or it's more common that the dynamic of vector-borne diseases, and especially of Culicoides, are just trying to understand in temperate areas. We say, okay, this is a summer species, this is a fall species, this is a winter species. So we use self-informed models to try to understand how the community could give you information about that natural dynamics. Yeah. So that was very interesting because in northern Florida we are expecting that the community behave like a temperate one. However, we have a picture that is a mixture between a subtropical and temperate behavior, because there is a mixture between dry and rainy summer, fall seasons. So we are wondering right now if that also means that the pattern of HDD will be something intermediate. It's like a continuum between the tropical and subtropical and temperate areas, and that's also that have been proven for other ecosystems. Then we trying to understand how different environmental drivers could assist us to understand and to predict when what different species will reappear. One of the most interesting things is that first, Culicoides in northern Florida are active year-round, so we don't have any gap that will be interrupted transmission. And also that the precipitation are not so important that we previously was mentioned. So we have two constraints that are related to the amount of groundwater and no precipitation, and also the temperature. The last thing that we can be working with Theo and Dinesh is try to understand how human-made hotspots for the transmission are led to the contact between vectors and hosts. So one of the main theory behind that is about the natural linearity of the transmission disease, or how super spread and super spreads and hotspot used to overlap in space and time. So we designed an experiment to try to understand how the food supplementation on deer could increase the chances of contact between Culicoides and host. So the first hypothesis that we have been trying is how food supplementation in feed stations facilitate Culicoides ruminant encounters. We already saw that, and we also saw that it's very interesting that that encounter is interacting strongly with the location of the feeder. So that means that could be we can make several recommendations of where is the right place, or where are the better place to allocate feeders and reduce the rate of encounter. So taking in consideration that, we started to continue with that experiment, trying to understand how the foraging theory can explain why Culicoides are exploring one feeder more than the others, and also how the food supplementation could increase and enrich the larval development sites, and that will be increase the chances of transmission, because the density of Culicoides also will increase. So the main idea with all of these subjects, I would say, is trying to make an ecological synthesis of what I have been working in the lab for the last 4 years with Culicoides. So that was all.
Hello, everyone. My name is Dinesh Erram, and I'm a postdoc with Nathan. A lot of you guys have probably seen me walk around with this hoodie on and thought, who's this weird guy walking around with FMEL hoodie on? But it's not that I'm that weird, but just get cold very easily. So I just leave this hoodie on in the lab, and whenever I come to the lab, I just wear this back on. So nothing weird about it, at least for me. Anyways, so so I'm broadly interested in the ecology, surveillance, and control of vector-borne diseases affecting human and animal health. So my postdoctoral work has mainly focused on hemorrhagic disease occurring in ruminants. So as Theo and Augustine pointed out, hemorrhagic disease is mainly caused by orbiviruses in the genus Orbivirus, the family Rheoviridae. This disease occurs mainly in ruminants, right? So mainly blue tongue virus and epizootic hemorrhagic disease virus are endemic in the United States. Blue tongue virus affects mainly sheep and cattle along with other ruminants, and EHD virus affects mainly white-tailed deer and cattle along with some other ruminants. However, with the recent findings of the cherry orbiviruses in white-tailed deer suggests that not just blue tongue virus and EHDV, or I mean, suggests that white-tailed deer are not just exposed to these two previously thought endemic viruses, but a whole variety of new orbiviruses. So the vectors of these viruses are mainly Culicoides species, commonly referred to as no-see-ums or biting midges, or sandflies here. However, cherry orbiviruses recently found are closely related to this Peruvian horse sickness virus or Mobuck virus that are mainly transmitted by mosquitoes. So we're beginning to suspect the role of mosquitoes also in the transmission of either of these orbiviruses. So that being said, why is this important? Is white-tailed deer farming is an important industry in the United States with a significant economic impact, right? So these viruses also affect not just white-tailed deer, but cattle and livestock in general. But white-tailed deer are the most affected ruminant species, demonstrating the need for research into this topic. So I'm interested in answering a variety of questions. Some of the questions that I'm interested in or have been working on as part of my postdoctoral work is identifying vectors involved in pathogen transmission. So previous work on orbivirus transmission in North America has been limited to Culicoides sonorensis, which is mainly found in the western states, but is largely rare in Florida or in the southeastern US, basically. So but we do find disease transmission occurring here. So it means that it's obviously other vectors involved in the transmission of these viruses. So we're trying to identify what species of Culicoides transmit viruses here in Florida, and then what host species are these vectors feeding on, like white-tailed deer, cattle, sheep, goats, whatever, what species of host these are feeding on, and then what are the habitats of these vectors, right? So we identified, so there's a picture from one of my studies where we identified or we tested 3 different habitats. So in A, we have a stream edge, B, we have seep edge, and C, we have a puddle area. So we've identified the puddle habitats and seep edge habitats to be mainly important in supporting the larval development of the main species. So we've identified Culicoides stellifer and Culicoides venustus as the likely vectors of virus transmission in Florida. And these species seem to occur in these manure or organically enriched substrates. Vector surveillance is another area of interest. My main questions that I am interested in answering is what are the cues involved in the main important life history traits of these vectors, right? So we have plant nectar feeding, we have host seeking, and then oviposition site selection as some of the main important life history traits. So what are the cues these vectors are queuing in on to complete these important life history traits, right? So there'll be long distance cues, short distance cues, visual, olfactory, tactile, which of these cues are mainly important in their life cycles. And then how do we exploit these cues in our surveillance and control strategies? But the ultimate question that I'm interested in answering is how do we establish vector control strategies, right? So we have cultural management strategies now that we have identified these habitats of the vectors and characterize their nutrient levels, macronutrients, micronutrients, pH levels, whatever. How can we manipulate these environmental variables so as to make these habitats unsuitable for vector oviposition and larval development, right? And then we have biological control methods, not quite established for Culicoides species, but we are trying to identify potential microbial agents that could be used as larvicides for these insects. and then the conventional chemical methods. Currently, no registered products are available for Culicoides species. So we're trying to test out different products for larvicidal and adulticidal uses, with the ultimate goal of incorporating all these knowledge into an effective integrated vector management program to protect our livestock. Thank you.
Hello, everyone. Today, I would like to share our progress of the automatic mosquito ID system we've been through. Let's get started. I have only 5 minutes, so I'm gonna be so busy. As you can see, that these multiple traps have been working so great for the mosquito surveillance. There is no doubt. But the problem is it is a very extremely time-consuming and labor-intensive that make the Dr. Burkett sweating a lot. We thought, what is the something better than before? As you can see the video, the mosquito flying like the swimming, that generated vibration from the air that as an acoustic wave. It is considered as a unique feature and the species specific. So since 1949, the many researchers failed to get this system into the real world for the mosquito classification. There are 3 main reasons. First, they can't handle the environmental noise from the microphone, very sensitive microphone. Second is poor sample size. Third is they use a very complex classification models with multiple parameters. Now, I would like to show you guys our magic mosquito ID system. So to get through all the previous limitations, we construct the apparatus with using the infrared, the emitter, and the receiver that automatically calculated mosquito wing beat. So this is literally the noise-free. So when mosquito pass through, that the sensor captures the mosquito flight movement that automatically calculated frequency, then it conveys to the cloud system. Very handy. As you can see the video, we released the single species mosquito inside of the apparatus and then let them chase the UV light that controlled by the timer. But this is the prototype. So they work so hard until they die. So we really appreciate it. He saved a lot of time. And then we built the mosquito, the wing beat profiling, and then from the 11 genera, 30 mosquito species that anyone never done before. We are so proud of ourselves. And then we also collect the additional wing beat profiling from the other non-target insects to increase our trap efficacy. Several systematic and structural modifications. We conduct the multiple experiments at the field and then semi-field. At first, we compare the trap efficacy. In updraft and downdraft, it is important for the trap design at the field. The results show that the updraft trap has a higher number of the mosquito capture. And then we compare the trap efficacy, the PG trap, with and without our system. Because it is important that our system can see if our system interfere the mosquito behavior or not. But the result was incredible. Our system in the single species setting, the highest recapture rate found in the 80 centile, 100%. And then our system comparing to the PG, the sentinel trap, but our system, the capture rate much higher than PG trap. We were so happy we beat the PG trap. Oh. And then in the gravity trap setting, so CDC gravity trap, a little bit much higher the recapture rate, but actually we improved our system from the 14% recapture rate to the 54%. We are still working on it. It'll be better. And then we conduct the field deployment and the validation study in the Florida and then Utah, see if our system really working at the field or not. So we set up the trap at the 3 different sites representing urban, suburban, rural, of course Utah. The consistent with the 3 different types of traps, the light trap, busy trap, gravity trap. And we found the 8 genera, 27 mosquito species in the Indian River County. So that is more than we expected. So, and then Utah, of course, we found the last mosquito species. The predominant species was Culicoides. And then we processed all the files we collect at the field. It was 0.5 million files. And then we tried to find what is the most reliable and then accurate the models. It was a multi-class decision forest. And then that accuracy to identify the mosquito was around the 60%. But if we add the more information, such as circadian rhythm and the geographical distribution, and then it go up around the 76%. We are still we collaborate the AI expertise in the Canada and the many engineers, so we can make it. So such an interdisciplinary approach can make it possible to provide the reliable, efficient tool to save the people. So the last one, the system, the benefit is that we can catch the mosquito alive without any damage. So thank you so much.
Right. Like everybody stayed on time. I think that's the last of it. This wasn't supposed to be like a real research talk, but if anybody's got questions for me or anybody else in the group, go for it. Yes.
When the Culicoides traps, how big do you have to go? Is it just
Over sampling the larvae?
Yeah.
That's these guys.
So the Culicoides larvae are generally in the soft of the water. So the deeper you go, the lesser.
The older you get, colder you're gonna get.
Now you know everything.